I am Seema, student of BSc Chemistry Honors Final Year. This video is especially for the visually impaired students, chapter Organic Chemistry chapter Enzyme and the Coenzymes. In these chapters, we study the general characteristics of the enzyme, enzyme structure, enzyme nomenclature, enzyme function, enzyme specificity, factors affecting the enzyme activity, enzyme inhibitions, regulation of the enzyme activity, medical uses of the enzymes. Firstly, general characteristics of the enzymes. Enzymes, usually a protein acting as a catalyst in the specific biochemical reactions. Each cell in the human body contains the 1000s of different enzymes. Every reaction in the cell requires its own specific enzymes. Most enzymes are globular proteins. A few enzymes are made of the RNA, catalyze biochemical reactions in nucleic acids. Enzymes undergo all the reactions of the proteins. Enzyme denaturation due to the pH or the temperature change. A person suffering high fever runs the risk of the denaturing certain enzymes. Enzyme structure. Simple enzymes composed only of the protein. Conjugated enzyme composed of the apoenzymes. Conjugated enzymes without its cofactor. Apoenzyme plus coenzyme gives to the conjugate enzymes. Apoenzyme, conjugate enzyme without its cofactors, protein part of the conjugated enzyme. The apoenzyme cannot catalyze its reaction without its cofactor. The combination of the apoenzyme with the cofactor makes the conjugated enzyme functional. Holoenzyme, apoenzyme plus cofactor, the biochemically active conjugated enzyme. Coenzyme, a cofactor, non-protein part of the conjugated enzyme. Coenzymes and the cofactor. Coenzymes provides the additional chemically reactive functional groups besides those present in the amino acids of the apoenzymes are either small organic molecules or inorganic ions. Metal ions often act as the additional cofactor. Zinc 2 positive ions, magnesium 2 positive ions, manganese 2 positive ions, and the iron 2 positive ions. A metal ion cofactor can be bound directly to the enzyme or to a coenzyme. Coenzyme, a small organic molecule acting as a cofactor in a conjugated enzyme. Coenzymes are derived from the vitamins or the vitamins derivative. Many vitamins act as the coenzymes, especially B vitamins. Enzyme definitions. The enzyme, protein only enzymes that facilitates a chemical reaction. Coenzyme, compound derived from the vitamin, example NAD positive, that assist an enzymes in the facilitating a chemical reaction. Cofactor, metal in ion, example magnesium 2 positive ions that assist an enzymes in facilitating a chemical reaction. Apoenzyme, protein only part of an enzyme, example isocitrate dehydrogenase, that requires an additional coenzyme to facilitate a chemical reaction, not function alone. Holoenzyme, combination of the apoenzyme and the coenzyme which together facilitating a chemical reaction. Enzyme nomenclatures. Enzymes are named according to the type of the reaction they catalyze and or their substrate. Substrate, the reactant upon which the specific enzyme acts. Enzyme physically binds to the substrate. Firstly, suffix of an enzyme is the ase, like lactase, amylase, lipase, protease, denotes an enzyme. Some digestive enzymes have the suffix in pepsin, trypsin, chymotrypsin. These enzymes were the first ones to be studied. Prefix denotes the type of the reaction the enzyme catalyzes. Oxidase means redox reaction, hydrolase, addition of the water to break one component into the two parts. Substrate identity is often used together with the reaction type. Pyruvate, carboxylase, lactate, dehydrogenase. Enzyme plus substrate then gives rise to the enzyme or the substrate complex. Enzyme class, reaction catalyzed, and the example in metabolism. First is the enzyme class oxidoreductase. The reaction catalyzed is the redox reaction, reduction or the oxidation. Examples. Example are the dehydrogenases catalyze the reaction in which a substrate is oxidized or the reduced. Transference, transfer of a functional group from the one molecule to another. Example is the transaminases which catalyze the transfer of the amino group, or the kinases which catalyze the transfer of the phosphate group. Hydrolysis reaction, lipase catalyzes the hydrolysis of the lipids, and the proteases catalyze the hydrolysis of the proteins. Addition and the removal of the atoms to from the double bonds. Decarboxylase catalyze the removal of the carboxyl group. Isomerization reaction. Isomerases may catalyze the conversion of the aldose to ketose, and the mutases transfer functional group from the one atom to another within a substrate. Ligase synthesis reaction, joining of the two molecules into the one, forming a new chemical bond coupled with the ATP hydrolysis. Synthesize link two smaller molecules or form a larger one. Enzyme acticides. Acticides, the specific portion of an enzyme location where the substrate bind while it undergoes a chemical reaction. The active site is a 3D crazy-like cavity formed by the secondary and tertiary structures of the protein part of the enzyme. Crevice formed from the folding of the protein. ACA binding cleft. An enzyme can have more than only one active sites. The amino acids means R group side chain in the active sites are important for determining the specificity of the substrate. Enzyme substrate complex. When the substrate binds to the enzyme active sites, an enzyme substrate complex is formed temporarily, allows the substrate to undergo its chemical reactions much faster. Lock and key model of the enzyme action. The active site is fixed with a rigid shape, means lock. The substrate key must fit exactly into the rigid enzyme block. Complementary shape and geometry between the enzyme and the substrate key substrate fits into the lock enzyme. Upon the completion of the chemical reactions, the products are released from the active site so the next substrate molecule can bind. Induced fit model of the enzyme action. Many enzymes are flexible and constantly change their shape. The shape of the active site changes to accept and accommodate the substrate. Conformations change in the enzyme's active site to allow the substrate to bind. Analogy, a glue enzymes that changes the shape when a hand substrate is inserted into it. Enzyme specificity. Absolute specificity, an enzyme will catalyze a particular reaction for only the one substrate. Most restrictive of all the specificities not common. Catalase has absolute specificity for the hydrogen peroxide. Urease catalyzed only the hydrolysis of the urea. Group specificity. The enzymes will act only on the similar substrate that have a specific fundamental group. Carboxypeptidase. Cleavage amino acids one at a time from the carboxyl end of the peptide chain. Hexokinase adds a phosphate group to the hexoses. Enzyme specificity. Linkage specificity. The enzyme will act on a particular type of the chemical bond irrespective of the rest of the molecular structure. The most general of the enzyme specificities: phosphatases, hydrolase phosphate esters bond in all the type of the phosphate esters. Chemotrypsin catalyzes the hydrolysis of the peptide bonds. Stereochemical specificity. The enzymes can distinguish between the stereoisomers. Chirality is inherent in an active sites as amino acids are the chiral compounds. L-amino acids oxidase catalyzes the reaction of the L-amino acids but not of the D-amino acids. Factor affecting the enzyme activity. Enzyme activity measures of the rate at which an enzyme converts the substrate to products in a biochemical reaction. Four factors affect the enzyme activity. One is the temperature, pH, substrate concentrations, enzyme concentrations. First is the temperature. With increased the temperature, means kinetic energy increases, more collision, increase the reaction rates. Optimum temperature is the temperature at which the enzyme exhibits the maximum activity. Optimum temperature for the human enzymes is 37 degrees Celsius. When the temperature T increased beyond the optimum temperature, changes in the enzyme tertiary structure occur, the inactivating and the denaturing its. Example, fever. Little activity is observed at the low temperature. pH. Optimum pH is the pH at which the enzyme exhibits the maximum activity. Most enzymes are active over a very narrow pH range. Protein and amino acids are properly maintained. Small changes in the pH, low or the high, can result in the enzyme denaturation and the loss of the function. Each enzyme has its characteristics pH optimum, which usually falls within the physiological pH range 7.0 to 7.5. Digestive enzymes are exceptions. Pepsin in the stomach, optimum pH is 2.0. Trypsin in the SIP, optimum pH is the 8.0. Substrate concentrations. If the enzyme is kept constant and the substrate is increased, the reaction rate increases until a saturation point is met. At the saturation, the reaction rate stays the same even if the substrate is increased. At the saturation point, substrate molecules are bound to all the available active sites of the enzyme molecule. The reaction takes place at the active sites. If they are all active sites are occupied, the reaction is going at its maximum rate. Each enzyme molecule is working at its maximum capacity. The incoming substrate molecules must wait their turn. Enzyme concentration. If the substrate is kept constant, the enzyme is increased. The reaction rate increases. The greater the enzyme, greater the reaction rate. Rule. The rate of an enzyme catalyzed reaction is always directly proportional to the amount of the enzyme present. In a living cell, the substrate is much higher than the enzyme. Enzymes are not consumed in the reaction. Enzymes can be reused the many times. Mechanism of the enzyme actions. Formation of an enzyme substrate complex as an intermediate species provides a alternative pathway with the lower activation energy through which a reaction can occur. Lock and key models, and second is the induced fit theory. Lock and key model, the active sites has a fixed geometrical shape. Only a substrate with a matching shape can fit into it. Induced fit model, the active site has the flexible shape that can change to accept a variety of the related substrates. Enzymes vary in their degree of the specificity for the substrate. Factors that affect the rate of the enzyme activity are temperature, pH, concentration of the substrate, and the concentration enzymes. First is the temperature. Reaction rate increases with the temperature until the point at which the protein is denatured and activity drops sharply. pH. Maximum enzyme activity is possible only within a narrow pH range. Outside this pH range, the protein is denatured and activity drops sharply. Concentration of the substrate. Reaction rate increases with the substrate concentrations until full saturations occur. Then the rate level of concentration of the enzyme. Reaction rate increases with increasing the enzyme concentrations, assuming the enzyme concentration is much lower than that of the substrate. Key concept: function of an enzyme. What is the function of an enzyme in a chemical reaction? What happens to the enzyme when the body temperature rises from 37 to 42 degrees Celsius? Enzyme inhibition. Enzyme inhibitor, a substance that slows down or stops the normal catalytic function of an enzyme by binding to the enzyme. Three types of the inhibition: reversible competitive inhibition, reversible non-competitive inhibition, irreversible inhibition. Reversible competitive inhibitions. A competitive inhibitor resembles the substrate. Inhibitor competes with the substrate for binding to the active sites of the enzyme. If an inhibitor is bound to the active sites, prevents the substrate molecules to access the active sites, decreasing or the stopping enzyme activity. The binding of the competitive inhibitor to the active sites is a reversible process. Add much more substrate to outcompete the competitive inhibitor. Many drugs are competitive inhibitors. Antihistamines inhibit the histidine decarboxylase, which converts the histidine to the histamine. Reversible non-competitive inhibitors. A non-competitive inhibitor decreases the enzyme activity by binding to a site on the enzyme other than the active sites. The non-competitive inhibitors alters the tertiary structure of the enzyme and the active site, decreasing the enzyme activity. Substrate cannot fit into the active sites. Process can be reversed only by lowering the non-competitive inhibitor. Example: heavy metals like lead ions, mercury ions bind to the SH of the cysteine away from the active sites, disrupt the secondary and the tertiary structure. Irreversible inhibition. An irreversible inhibitor inactivates an enzyme by binding to its active site by a strong covalent bond, permanently deactivates the enzyme. Irreversible inhibitors do not resemble the substrate. Addition of the active substrate does not reverse this process, cannot be reversed. Chemical warfare, nerve gases, organophosphate insecticides, enzyme inhibitors. Substances that bind to the enzymes and stop or slow its normal catalytic activities. Competitive enzyme inhibitors, non-competitive enzyme inhibitors, irreversible enzyme inhibitor. First is the competitive enzyme inhibitors, a molecule closely resembling the substrate binding to the active sites and the temporarily prevents the substrate from the occupying it, thus blocking the reactions. Non-competitive enzyme inhibitor, a molecule that binds to a site on enzymes that is not the active sites. The normal substrate still occupies the active site, but the enzyme cannot catalyze the reaction due to the presence of the inhibitor. Irreversible enzyme inhibitor, a molecule that forms a covalent bond to a part of the active sites permanently, preventing the substrate from the occupying it. Allostatic enzymes. Allosteric enzymes have a quaternary structure, are composed of two or more the protein chains, possess the two or more the binding sites. Two types of binding sites. One binding site for the substrate is the active site. Second binding site for a regular molecule is the regulatory sites. Binding of a regulator molecules to its regulatory sites causes the changing in the three-dimensional structure of the enzyme and the active sites. Binding of a positive regulator upregulates the enzyme activity, enhance the active sites more able to accept the substrate. Binding of a negative regulator non-competitive inhibitor down the regulates enzyme activity, compromise the active sites less able to accept the substrate. The different effects of the positive and negative regulations on the allostatic sites. Negative allostatic controls the substrate, means the allostatic sites, negative regulators, and then the active sites. Feedback control, a process in which the activation or the inhibition of one of the earlier reaction steps in a reaction sequence is controlled by a product of this reaction sequence. One of the mechanisms in which the allostatic enzymes are regulated. Most of the biochemical process proceed in several steps. Each step is catalyzed by a different enzymes. Example: the degradation of the glucose through a metabolic pathway can be regulated in several ways. The enzyme PFK is allostatically inhibited by the product ATP. Glycolysis makes the ATP is slowed when cellular ATP is in the excess. Proteolytic enzymes and the gymogens. Second mechanism of allostatic enzyme regulation, production of an enzyme in an active form activated when the required right time and the place activated eka turned on. Proteolytic enzymes catalyze the breaking of the peptide bond in the proteins. To prevent these enzymes from destroying the tissues that produce them, they are released in an inactive form. Gymogens. Most digestive and blood clotting enzymes are the proteolytic. Blood clotting enzymes break down the proteins within the blood so that they can form the clot. Platelets interspread with the tangent protein collagen and the thrombin. Activation of enzymogen requires the removal of a peptide fragment from the enzymogen structure, changing the three-dimensional shape and affecting the active sites. Example: pepsinogen. It is the covalent modification of the enzyme. Covalent modifications are the third mechanism of the enzyme activity regulations, a process of altering the enzyme activity by covalently modifying the structure of the enzyme, adding or removing a group to remove from the enzymes. Most common covalent modifications is equal to the addition and removal of the phosphate group. Phosphate group is often derived from an ATP molecule. Addition of the phosphate phosphorylation is catalyzed by a kinase enzyme. Removal of the phosphate dephosphorylation is catalyzed by the phosphatase enzyme. Glycogen synthesis involved in the synthesis of the glycogen deactivated by the phosphorylation. Glycogen phosphorylation involved in the breaking down of the glycogen activated by the phosphorylation. Vitamins as the coenzymes. Many enzymes require the B vitamins as the coenzymes allow the enzymes to function. Coenzymes serve as the temporary carriers of the atoms or the functional group. Coenzymes provide the chemical reactivity that the apoenzymes lacks. Important in the metabolism reaction to release the energy from the foods. Example: redox reaction where they facilitate the oxidation or the reduction. Vitamin B do not remain the permanently bonded to the apoenzyme. After the catalytic action, the vitamin is released and can be repeatedly used by the various enzymes. This recycling reduces the needs for the large amount of the vitamins B. Next is the drugs inhibiting the enzyme activity. Many prescription drugs inhibit the enzymes. ACE inhibitors inhibit the angiotensin converting enzymes, lowers the blood pressure. Sulfa drugs, antibiotics, acting as a competitive inhibitors of the bacterial enzymes involved in the conversion of the PABA to the folic acid. Deficiency of the folic acid retards the bacterial growth, eventually killing them. Penicillins. Beta-lactam antibiotics inhibits the transpeptidase. Transpeptidase enzyme strengthens the cell wall, forms the peptide crosslinks between the polysaccharide strands in the bacterial cell walls. Without the transpeptidase enzyme inhibited by the penicillin, greater than the weakened cell wall, bacteria dies. Medical uses of the enzymes. Enzymes can be used in the diagnosis and the treatment of the certain disease. Lactate dehydrogenase LDH is normally not found in the high levels in the blood as it is produced in the cell. Increase the level of the LDH in the blood indicate microcardial infarctions and heart attack. Tissues plasminogen activator TPA activates the enzyme plasminogen that dissolves the blood clots used in the treatment of the MI. There is no direct test to measure the urea in the blood. Urease converts the urea into the ammonia, which is easily measured and is used as a urea indicator. Blood urea nitrogen BUN is used to measure the kidney function. High urea levels in the blood indicates the kidney malfunction. Isoenzymes. Isoenzymes catalyze the same reaction in the different tissues in the body. Example, lactate dehydrogenase LDH consists of the 5 isoenzymes. Each isoenzyme of the LDH has the same function, converts the lactate to the pyruvate. LDH1 isoenzymes is more prevalent in the heart muscles. LDH5 form is found in the skeletal muscles and the liver. Isoenzymes can be used to identify the damaged or the diseased organ or tissues. It is a marker for a particular location. If the LDH1 isoenzymes was found in the blood is greater than indicates the heat muscles damage. Lactate reduced substrate plus oxidized coenzyme like NAD positive. In the presence of the lactate dehydrogenation, equilibrium is shifted and pyruvate oxidized product formed and the reduced coenzymes. Selected blood enzymes assess the used in the diagnostic medicine. First is the enzyme is the lactate dehydrogenase LDH. Conditions indicated by the abnormal level is the heart disease, liver disease. Creatine phosphokinase CPK, heart disease. Aspartate transaminase AST, heart disease, liver disease, muscles damage. Alanine transaminase ALT, heart disease, liver disease, muscles damage. Gamma glutamyl transpeptidase GGTP, heart disease, liver disease. Alkaline phosphate ALP, bone disease, liver disease. Next table is the selected important coenzymes in which the in which the B vitamins are present. Vitamin B act as coenzymes and the group transports thymine. Coenzyme is the thymine pyrophosphate TPP. Group transferred is the aldehydes. Riboflavin coenzyme is the flavin mononucleotide FMN, flavin adenine dinucleotide FAD, hydrogen atoms. Niacin nicotinamide adenine dinucleotide NAD positive, nicotinamide adenine dinucleotide phosphate NADP positive, hydrogen atoms. Pantothenic acids coenzyme A acyl groups. Vitamin B6 pyridoxal 5-phosphate PLP, pyridoxine 5-phosphate PNP, pyridoxamine 5-phosphate PMP. Amino group. Biotin carbon dioxide carboxyl group. Folate tetrahydrofolate THF 1 carbon group other than the CO2. Vitamin B12 methyl cobalamin methyl groups, hydrogen atoms. Thank you. This is all about the enzymes and the coenzymes.